everybody. Woody is going to start recording our meeting. So just to let everybody know, you are now being recorded. Welcome to the Monday Night Speaker Series. And Woody and I are the co-directors for Washington State of NAME. I'm Jo Vanina.
I'm Woody.
That's Woody. And NAME, for those of you who don't know, is an organization of educators, marine and aquatic educators from Oregon, Washington, British Columbia, and Alaska. And we're very happy that you are here tonight to join us. We have Dr. Louise Page, who will be talking about astonishing annelids. And just a reminder for everybody, June 14th will be our next talk and the last one for a couple of months. We're going to take a break over the summer. But
wasn't there one in May?
Oh, we have one in May. Dang it. Oh, crap. Okay, sorry. No, we have one in May before June. I thought it was May already.
The sunshine comes out. Yeah, the next talk is going to be.
May. What is the date on that one?
Yeah. It's going to be on seabed mining.
Sorry.
It's all good.
And then there's the June 14th one.
Right.
But the May one is on the 10th. May 10th. Sorry, I got ahead of myself. But yeah, thank you all for coming tonight, and I'm gonna stop my share. But if anybody has any questions about NAME, just go ahead and let me know, and then we can get started.
I think Kathy was going to introduce tonight's speaker.
Yeah, this is Kathy. I'm past president of NAME, the Northwest Aquatic and Marine Educators. And I'm super, super stoked and happy that Louise was willing and enthusiastic about presenting tonight. She's been teaching and researching about invertebrates for more than 40 years now. And she has lots of great stuff to share. So welcome everybody, and thanks, Louise.
Thank you.
Well, thank you. What a wonderful introduction. Kathy and I have known each other for a long time. So hi, everyone, and welcome to Amazing Annelids. And I just want to say thanks to the Northwest Aquatic and Marine Educators for putting on this virtual speaker series. I know I very much enjoyed the presentations so far. But as Kathy said, I'm Louise Page, and I'm an associate professor at the University of Victoria. So that's on Vancouver Island. And so I'm an educator. And needless to say, this past year has been really quite traumatic to be an educator, because we suddenly had to deliver learning materials online. And honestly, I really take my hat off to those teachers that are teaching primary schools and secondary schools, because that must have been a real challenge. I can't imagine how they managed it. But I teach a course in invertebrate biology, and it's directed toward third-year undergraduates, but I also have second and fourth-year undergraduates in the course. And the course consists of 3 hours of lectures every week and a 3-hour lab. So there's about 9 labs during the year. And each one is focused on a different group, a different phylum of invertebrate. And the beauty of teaching invertebrate biology at the University of Victoria is that I can access live marine invertebrates within literally walking distance of the university. So typically the labs consist of live invertebrates that we've collected locally. We bring them into the teaching lab, and then UVic has a recirculating seawater system. So when the students come into the lab, they can observe and study the morphology and the behavior of live marine invertebrates. And we also do a few dissections. So all of a sudden, a year ago, we had to rapidly figure out how to deliver a facsimile of this sort of hands-on self-exploration type of learning experience onto an online platform. So some rapid decisions had to be made, and I decided that I would create about 3 videos for each of the 9 labs, illustrating various different aspects of the group under study. So showing the students what they might expect to see if they were attending the lab in person. And then my very dear and old friend, Kathy Karelsfeld, after it was all done, thought that one of these videos might be of interest to this group. So what I'm going to do is I'm going to present just one module within the annelid lab, and this is marine annelids, segmented worms. It was the module focused on feeding diversification within the marine annelids. So that's what I propose to show you today. And I've tacked on a little video right at the beginning because I want to introduce you to some terms that I'm going to use repeatedly throughout all the videos. And I want to make sure that everybody understands what the meaning of those terms are. So finally, I want to say that all of the animals that I'm going to show you, 4 major groups, they were all collected from the intertidal within the traditional territory of the Lekwungen speaking First Nations people. So with that, I'll launch this first video. I'll have to share my screen.
Hey, Louise.
Yes?
I was just going to remind people that we will watch the chat. So if you have questions, put it in the chat. And then when we come to a break, I'll ask the questions, or Woody will ask the questions of Louise.
Thank you very much for remembering to say that.
Okay.
So this is the first little introductory video clip, and it's just going to introduce you to some terms about marine annelids. Okay, what you're looking at here is a large marine annelid within the family Nereididae, commonly known as ragworms or clamworms. And they look like large aquatic centipedes or millipedes because the body consists of a long linear series of segments, which we call metameres. And each metamere has a pair of little legs coming off each side, one on the left and one on the right. And these legs are called Peripodia. So you're looking at a Nereidid worm using its Peripodia to crawl over sandpaper covering the bottom of a marine aquarium. So notice the stepping movements of the Peripodia. Now these worms are also very capable of getting up off the surface and swimming, during which the Peripodia function as swimming paddles, and their paddling motions are supplemented with lateral sinusoidal undulations of the body. So I'd just like to say a bit more about a Peripodium. This is a Peripodium that's been isolated from the worm's body. And you'll notice that each Peripodium consists of 2 lobes. So there's a large upper lobe, a dorsal lobe that we call the notopodium. And then there's a smaller lobe on the ventral side, or the lower lobe, and it's called the neuropodium. And both of these lobes have a fan of stiff bristles projecting out from them. And these bristles are called chaetae. So the chaetae increase traction during crawling, or they also increase the surface area of the Peripodium when they're being used as swimming flippers. Okay, so just one last thing here. There's 2 main groups of marine annelids, the Aurantia and the Sedentaria. So the Aurantia, and this group includes those Nereididae that I've just been talking about, these are the active marine annelids, the so-called wanderers. So these are, some species are active burrowers, others are active crawlers, and many of them are capable of swimming. The Sedentaria, on the other hand, are the stay-at-home marine annelids. So they live in permanent burrows within the sediment, and by that I mean mud and sand, or they live in tubes that they construct. So I'm going to talk about feeding in 4 species of marine annelids. One is an Aurantid, and that is a bloodworm. And I'll talk about 3 different groups of sedentarid marine annelids, the lugworms, the spaghetti worms, and the feather duster worms. Okay, so at this point, I'll say, does anybody have any questions? That was all pretty straightforward stuff, I think.
We had one request about the audio from the video clip. If you think that the audio is necessary, I think it'd be fine to leave it on. But if you're talking over it, and it's just sort of that bubbling sound in the background, it was a bit hard to hear before.
Okay, that might not be so much of a problem later. That bubbling sound was because I recorded the video with the animal in a sea table,
Okay.
and the inflowing seawater was making that bubbling sound. So that won't be superimposed on all the subsequent videos.
Okay, cool.
Hopefully that won't be a problem going forward. Okay, well then I'll launch the next video then, which is going to be about feeding and burrowing behavior for the group commonly known as the bloodworm. So the proper name is Glyceridae. So with that, okay, this is me back in the days when we could get haircuts. So with that, onward to the glycerids. This is a glycerid, otherwise known as a bloodworm. The worm is folded back on itself, so this image shows only the anterior and posterior ends. But it doesn't matter because everything in between looks very similar. A bunch of metameres, each with a similar pair of peripodia. The peripodia are smaller than those of nereidids. The prostomium of bloodworms is distinctive. It's a pointed cone with 4 tiny antennae at the tip. At the posterior end, the pygidium bears a pair of so-called anal cirri. An enlarged view of the anterior end shows the conical prostomium and 4 tiny antennae. The red color of these worms is due to hemoglobin contained within coelomocytes, which are cells that float around within the coelomic fluid. These worms don't have a blood vascular system, nor do they have septa at the metamere junctions. This drawing shows a cutaway view of a block of sandy beach, showing the underground gallery of tunnels built by a species of bloodworm, with the bloodworm itself hanging out with its head just below the surface opening of one of the tunnels. These worms are predators. When a prey item, like a little ostracod or tiny juvenile crab, wanders too close to a tunnel opening, the enormous tubular pharynx of the bloodworm is shot out. The everted pharynx has 4 hollow jaws, each provided with a duct from a venom gland, and the prey is rapidly immobilized. But just like many nereidids, the aversible pharynx has 2 functions in glycerids. Not only does the pharynx function for feeding, but it also provides the primary, if not the only mechanism for burrowing into sediment. Bloodworms do not swim or even surface crawl with much proficiency. They basically just coil up or thrash around when they're on a surface, occasionally everting the pharynx in an effort to dig in. This is shown in the following video clips. I have 2 species of glycerid to show you, one much larger than the small species that I've been showing you so far. This large glycerid was found under a rock at Telegraph Bay, where this video was recorded. Notice how it probes around with its long, flexible, pointed prostomium. Now, wait for it. Yikes. Here is it performing again. Kind of scary. Here's the smaller species of glycerid. It looks almost cute after the previous big bruiser of a glycerid. It's on a piece of sandpaper with a bit of real sand on the surface. Poor frustrated bloodworm. This is just a slow motion clip of that previous video clip. And finally, here's the glycerid on a piece of wet countertop cover. Okay, well, I've shown you a bunch of video clips of the pharynx being used for attempts to burrow, but what about its use in predatory feeding? I'm sorry, but I do not have videos of that. However, I'm just sure that you love to take a look inside one of these worms to see the nasty jaws and their associated venom glands. I know that I'm keen to expose these jaws, so I'm going to undertake a dissection for this purpose. But before I do, I want to point out that, at least for the smaller species of glycerid, you can see the position of the jaws even within an intact live worm, because the jaws are black and the body wall is a bit see-through. They're black because one of the structural materials forming the jaws is melanin. Melanin is used for a lot of things by animals, but I don't normally think of it as a structural material. Nevertheless, the jaws also incorporate copper, both in mineral and in non-mineral form. There is evidence that the non-mineral copper may act to form crosslinks within and between melanin molecules, therefore hardening the melanin. Okay, well, what I'm doing here is I'm getting set up to do a dissection of a glycerid annelid, commonly known as a bloodworm. So the first thing I have to do is get the worm and then anesthetize it. So I'll go to the C table and get the worm out of the C table. Okay, well, here it is. As you can see, these things are not much of a swimmer. They don't generally swim. Okay, so to anesthetize it, what I'm going to do is I'm going to put it in an artificial seawater solution with a minimal amount of calcium and an overabundance of magnesium ions. This interferes with synaptic transmission, so that's what I'm going to use for anesthesia. So I'll pour some into this bowl here, and then I'll transfer the worm from the normal seawater to the artificial seawater. Trouble is, I'm going to be careful with it because they do have that potential to bite and inject their venom. So I'll just use this little mini petri dish to transfer it over to the bowl of artificial seawater, high magnesium, low calcium. So I'll put that in the fridge for a couple of hours. In the meantime, I want to show you the tools I'm going to use to do this dissection. I'm going to use a glass petri dish. The bottom of the petri dish is covered with a layer of material called Silgard. This is great stuff. You make it up, you mix two solutions together. Initially it's a liquid, and you pour that liquid into the bottom of a dissecting dish, and then the resin polymerizes. So the nice thing about Silgard is it's transparent. So if you want to do your dissection on a dissecting microscope, you can illuminate it from underneath, which is useful in some situations. It's also very good at taking little tiny pins. So I'll use a Silgard Petri dish for my dissecting tray. And then I've got a couple of pairs of fine forceps. I've got a third pair of forceps that are really fine. This is a very small animal, so it's going to require delicate dissecting tools. And then I've got a pair of watchmaker's scissors. You just close them by depressing these metal flanges. These are very useful for doing fine dissections under a dissecting microscope, and that's what I'm going to be doing. And finally, of course, we've got some very tiny minution pins for pinning out the body wall. I'm going to be making an incision up the dorsal side of the worm, just to one side of the mid-dorsal line, and then I'm going to be using these minution pins to pin back the body wall. Okay, well, I'll put this animal in the fridge for a couple of hours and it should be thoroughly anesthetized. I'm partway through the dissection, so I wanted to show you the initial incision along the dorsal body wall, with the body wall pinned to either side. I'm showing you this posterior to anterior. This is the midgut covered with yellow chlorogogen cells that perform liver-like functions. But my incision hasn't yet gone far enough anterior to reach the black jaws, so more cutting is necessary. I've now completed the incision all the way to the anterior end, and I'm scanning down the length of the pharynx from anterior to posterior. Note the longitudinal folds in the wall of the pharynx, which allow it to expand. And then, voila! At the base of the pharynx, you can see 2 of the 4 jaws through the transparent wall of the pharynx, and you can see the associated venom glands. Continuing posteriorly, note the folded esophagus. It needs to be folded so that it can unfold when the pharynx is everted. And then the midgut, covered in yellow chlorogogen tissue. Here's the jaws and venom glands, abbreviated VG, at the base of the retracted pharynx that I exposed with this dissection. An interesting study was done a few years ago in which the procedure known as in situ hybridization was used to localize the site of venom synthesis. Surprisingly, synthesis was not in the venom glands, but rather in these little pharyngean lobes indicated by arrowheads. The authors concluded that the so-called venom glands were actually just bags for venom storage. Okay. So that was the video on glycerids, bloodworms. And I'll stop here to answer any questions that you might have. I see there's 4 in the chat.
Yeah.
We have 2 questions. One is, do nereids have jaws made of the same thing, melanin and copper, or is it specific to glycerids?
No, the melanin and copper, well, the melanin is specific to glycerids. Nereids also have jaws, but they have only 2 jaws. They've got a pair of jaws rather than 4. And the jaws are formed of a chitinous material in nereids. I think they harden the jaws with a biomineral, and I should remember what that biomineral is, but I can't remember. I think it might be copper, so biomineralized copper. It could be zinc, but they do use a biomineral to help harden the jaws, the nereids. But they're a much larger jaws, and there's only 2 of them in nereidids.
And they're made of chitin, you said, a chitinous material?
Yeah.
Okay. And then the second question was, how toxic, and I'm guessing they're talking about the venom, are these worms, and would it cause a problem to a to a human?
No, it would probably cause a little sting, but they're not injecting very much venom. But certainly it's enough. It's an anesthesia. So its effect on other organisms is to rapidly disable them so that they can be captured.
Wow.
But you know, they're too small to really be dangerous. Well, the toxin isn't potent enough and the worms don't deliver enough of it to really be any more than an irritant to a human.
Okay. Would the jaws puncture skin?
I think they could be. Kathy, have you ever been bitten by a bloodworm? She's been stabbed by Nereids, by nematines, I know that.
Yeah, the nematines are the one that surprised me, but I have been jabbed by a glycerid, but it wasn't nearly as bad as a nematine.
Okay, there you go. Expert.
And then there's another question. It says, below the stomach was something that looked like a tadpole. Did I see that correctly?
Below the stomach, I didn't actually show you the stomach. Was it the midgut? Oh, no, it wasn't. Definitely wasn't a tadpole. There was some air bubbles forming in the water because the water was warming up. I wonder if it might have given you that impression.
Okay.
I'm not really sure what to say there.
And then another question, what type of critters do they eat?
Oh, all kinds of things. Small-sized organisms, as I said, like little crustaceans like Teneids, or small little newly metamorphosed crabs, ostracods, small worms. Yeah, they'll feed on a variety of things, provided they're small enough, and they detect the vibrations, so they know when to shoot out their pharynx.
Okay. And the fawn was the one who asked the question about the tadpole looking structure. She said it wasn't a tadpole, it was just, it looked like a structure in the body that was that shape.
Okay, I'm sorry, I'm just, I don't want to go back to it because we'll run out of time.
Okay.
But I can say for sure it wasn't a tadpole.
Yeah. All right, that was all the questions for this section.
All right, then I'll share my screen again. And I will show the next video clip. This is the first of 3 video clips. It's going to be about a member of the Sedentaria. So these lugworms live in permanent burrows within the sediment. And just so you know, sediment means mud and sand. And things that eat sediment are called deposit feeders.
You've seen this beast before.
Oops, I'm sorry.
It's Abernicola, a member of the family Abernicolidae, commonly known as lugworms. I described the morphology of Abernicola when I chose it as a representative sedentary annelid, but now I've put together some images and video clips to help you understand the lifestyle and feeding method of lugworms. Lugworms live in burrows in muddy sand, and they're bulk deposit feeders. They eat sediment particles and digest off the organic surface film. This sketch is a cutaway view of a block of sandy benthos, showing a lugworm in its burrow. The burrow is J-shaped. It has an opening at the surface of the sand, but the worm lives with its anterior end at the blind end of the burrow. Lugworms are very capable of peristaltic body contractions, but unlike in earthworms, where body peristalsis is used for burrowing, in lugworms, body peristalsis pumps seawater into the open end of the burrow. The water flows through the burrow, then percolates up through the sand above the blind end of the burrow. The worms eat sand grains at the blind end of the burrow, which is continuously caving in, often producing a sinkhole in the sand above the head end of the worm. Water pumped into the burrow by body peristalsis helps bring in oxygenated surface water to flow over the gills, which contain hemoglobin. But a happy side effect of having this water constantly flowing through the burrow is that unicellular algae, diatoms, and small zooplankton in the overlying seawater gets filtered out by the sand at the blind end of the burrow, thereby enriching this area of sand with organic matter, which gets eaten by the lugworm. But how can I be so confident about lugworm behavior, and how can I convince you that it's true when lugworms live buried in sediment that no one can see into? Well, lugworms excavated from their burrow will accept a glass pipette as a substitute burrow, and of course we can see through a glass pipette and examine what the worms are doing under a dissecting microscope. So here's a lugworm in a glass pipette, and you can definitely see the peristalsis. The peristaltic wave is traveling toward the anterior end. This was filmed in real time. This is more peristalsis, but I want you to also note that these worms have very modified peripodia. The notopodial part has a long brush of chaetae, but the neuropodium is just a slit bordered by raised lips, and the lips bear minute chaetae called uncini. Okay, well, there are many possible ways of ingesting sediment particles. How do lugworms do it? Well, they have a large bag-like eversible pharynx covered with sticky papillae. When the pharynx is everted, sand grains get caught in the spaces between the papillae, and when the worm draws the pharynx back in, it pulls in the sand grains also. The worm then digests off the organic surface film surrounding the sand grains. But I'd like to bring this alive for you, literally, by showing you the lugworm in the glass pipette everting its pharynx covered with papillae. So here it is. Although the lugworm is perhaps a bit confused because I haven't put any sand into the pipette. But I hope you can appreciate that sand particles can get trapped between the small papillae of this pharynx. It's so fun to watch. The worm needs to stabilize its position within the burrow while it's feeding, and it does this by gripping the sides of the burrow with the chaetal brushes of the notopodia. But the tiny hook-shaped chaetae of the slit-shaped neuropodia also act as little grippers. Nevertheless, the much longer chaetal brushes of the notopodia can also act as little legs to allow the worm to crawl up and down in its burrow. And this is important because periodically the worm needs to back up in its burrow and defecate all the sand that it's eaten, now cleaned of organic matter. And indeed, you can locate a lugworm living below the sand of a patch of beach because there's a little mound of fecal castings beside the open end of its burrow.
Okay, well, that was the lugworm story. Honestly, I had so much fun collecting these video clips. I mean, teaching shouldn't be this much fun. Anyway, you specialize on a particular group of invertebrate. For me, it's gastropod larvae, but I teach about all these different invertebrates. And yet, how often do you really have time to carefully look at their morphology and what they're doing? So this is really fun for me. Anyway, any questions about lugworms?
We did have a question. Justin was asking how deep the burrows are.
Oh, the burrows, okay, they would be no more than about 5 inches, I think. 3 to 5 inches, I'd say.
3 to 5.
The worms are only you know, they're only about 1.5 inches long. So depending on the species, they would, so I'm thinking more like 3 inches down burrows. Any other questions?
Can I ask a question by mouth?
Yes, absolutely.
So we've noticed with Abernicola that there's really big populations in certain places, but they really come and go. Like sometimes they'll be really common in one little area 1 year, and then they're gone for 5 years, and then suddenly they're back again. Do you?
Kathy, I've noticed exactly the same thing. Oak Bay mudflats, for example. Some years I don't see any. This last June, they were everywhere. So how to explain that? I mean, just the phenomenon, when you go to a sandy beach, a long stretch of sandy beach, it looks absolutely uniform, scanning it from the surface in general. And yet you find sand dollars only in this patch here. And then you find Abernicola in another little patch, and then maybe moon snails over here. So why is this their differential distribution across a beach that looks so uniform? Periodic occurrences between years. There could be another explanation. Sometimes, and I'm just speculating here because I don't actually know the answer to this question, but a number of species of marine invertebrates, the larvae are induced to settle by organic effluent from the adults. So when there's a large population of adults, you'll get lots of recruitment of larvae into that area. And so you'll get a large population built up. But if there's a few years of no recruitment, then, you know, the adult population may die out, and it may take more time for the population to become reestablished again. So it could be some sort of phenomenon like that. That seems to be true for sand dollars at any rate. But again, I'm just waving my arms around here because I don't know exactly in the case of lugworms.
Great, thanks.
We had a question about the oxygen needs for the lugworms, how much they require.
Well, when you think about it, these things are living buried in the sediment. And if you go to a beach like Pat Bay in the summer, if you dig down just a little bit under the surface, you get this hydrogen sulfide smell. And you know that that level of the sand is anoxic. And so the sulfide is produced by anoxic bacteria. So any worm that lives within the sediment, very often they have hemoglobin in their blood or their coelomic fluid, and that's to help pull out this oxygen from the surrounding seawater because the oxygen partial pressure is very low. The poor water buried in the sediment, the oxygen partial pressure is very low there. So they need to have hemoglobin to help pull out what oxygen is available.
Excellent. Patricia asked if you could talk a little bit about how you film the videos.
Yeah. I bought an iPhone. And the reason I bought an iPhone, so that's why these are not professional, is I was just telling the host a little bit earlier, Woody, that I taught a course at Banfield Marine Science Center a few years back. And at the time, I just had an ancient Samsung myself, but I just couldn't believe the quality of the images and the videos that these students taking the course were obtaining. So as soon as I discovered that I was going to have to create a bunch of videos for an online lab, I went out and bought myself an iPhone. And then I got a video editing software that I learned how to use through YouTube videos. And this is called Movavi, M-O-V-A-V-I. There's a lot of free video editing software out there, but I'm really happy with Movavi. It was fun to use. And there was a lot of trial and error, believe me, a lot of patience, but it was all fun.
Excellent.
I also had, of course, cameras mounted on a dissecting microscope. And I also had a student microscope, a compound light microscope that had a video camera on it. So I did have those as well.
Can you hear us at this point?
Yes, I can. Can you hear me?
Yes, I can. Okay. I have a question that is a little bit off your topic, but not really. Talking about the Nereis, could you talk a little bit about their swarming behavior when they reproduce? And like I've heard kind of different stories, but that they use their jaws to cut each other into pieces and blah, blah.
Oh, I don't think so.
Well, that's what I'm
No, they, so every, once a year at temperate latitudes, some of the errantid species of marine annelids, including Nereids, they're separate male and female individuals, and they've all developed these gametes. So this is their genes that they need to pass on. And what they do in response to an environmental cue, and it's usually a lunar clue, they'll all rise up into the water column doing that swimming behavior, and they all release their gametes, the eggs and the sperm. So this behavior, which is called swarming,
right, yeah.
is a mechanism to ensure that eggs and sperm are together at the same time in the same place to facilitate successful fertilization.
Okay.
So, I mean, I could talk on about this, but then I'll not have time
Right.
to show you my next two videos. So Gloria, certainly I'd be happy to talk about that more next time we see each other.
Okay, good.
Well, I have a question too. Can you hear me?
Yes, I can.
Okay, I'm a Giovanni, who's not.
Well, that's my wife. She's
Okay.
I'm still here. Yeah.
All right. The bloodworms, do they get called bloodworms because they have hemoglobin, because they live down in the anaerobic layer of
That's
sediment?
exactly right. Same reason that the lugworms have hemoglobin.
Ah, okay, thank you.
And we're going to see more hemoglobin as we go along here.
Our dog
Okay,
Turtle is interested too. She keeps coming here.
I'm going to just go on to the next video. Time is, so let's see. Okay, this is Terabellid. So this is the spaghetti worms.
Manja.
Here's a second example of a sedentary annelid. You're looking at the long, white, very mobile tentacles that extend from the head of this worm. The rest of the worm's body is encased in a sand tube on the underside of the rock. The anterior end also gives rise to curly red gills. They are red because of hemoglobin in the blood. This is a member of the annelid family Terabellidae, commonly known as spaghetti worms, for obvious reasons. Spaghetti worms may ingest sediment particles occasionally, but primarily they deploy their long feeding tentacles to creep over the substrate to pick up detritus from benthic surfaces surrounding their tube opening. The tentacles also pick up sediment particles and other materials, which are transported back to the head of the worm and used to build their tube. Many terabellids build their tubes out of whatever is available. The individual that built this tube within a large 2-liter glass bowl had sand available, but often the tubes incorporate small pebbles and fragments of mollusk shell. An adhesive is secreted to consolidate all these materials into a cohesive tube, which is open at both ends. This is an image of the same tube where it climbed up the side wall of the bowl. You can see the central channel of the tube because the worm used the transparent glass wall of the bowl to form part of the wall of its tube. At the time this image was taken, the worm had retracted away from the tube's opening, and only slender tentacles can be seen swirling about inside the lumen of the tube. I was hoping that the worm would eventually crawl up the side of the bowl so that I could see what it was doing in its tube. But unfortunately, the worm turned around in its tube and started building up the other end, but I still couldn't see it from the side wall of the bowl. So I needed to photograph the worm from the bottom of the bowl. Bit of a technical challenge. So what I did was prop the bowl between a desktop and a close-by tabletop with a gap in between. I put a lamp underneath and then crawled under the table to video the bottom of the bowl where the worm was located within its tube. So here's me crawling under the table. But initially, the worm didn't seem to be doing much of anything in its tube. But after a short while, it started with peristaltic contractions of its body. These peristaltic contractions are not for burrowing, but rather they function to pump seawater through the tube in order to keep the gills bathed in oxygenated seawater. Remember, these annelids typically build their tubes under rocks resting on sand or mud, where oxygen partial pressures can become low. Body peristalsis is also used to move along the length of the tube. You've been looking at the large local species of terabellid, Philippus insignis, and here I've removed one from its sand tube. The peripodia of this robust worm are much modified and function primarily as grippers to hold onto the inner wall of the tube. But how do those long white tentacles pick up sediment particles and surface detritus? This is a view of a feeding tentacle observed under a dissecting microscope. As the tentacle twists slightly about its long axis, you can see that the tentacle has a deep groove running down its length. Furthermore, these tentacles are
I
ciliated.
need to stop this right here to explain what cilia are. Cilia are microscopic whip-like appendages that beat back and forth very actively, and there's thousands of them on the surface of animals. And so they can propel an animal over a surface like a crawling slug, or they can direct seawater past an animal, or they can carry particles along an animal, which is what's happening here. So I'll butt out now.
The cilia allow the tentacles to creep over the substrate, and the ciliated groove picks sediment particles and detritus for transport back to the head for tube building or ingestion. This movement of particles along the ciliated groove can be seen in the following two videos. The fact that some of the particles transported back to the mouth along the tentacles are used to build up the tube, whereas others are ingested, suggests that terabellids have a sorting mechanism for incoming particles.
Okay, I'm going to stop it there and ask for any questions about the spaghetti worms.
So,
Sorry.
Louise, this is Kathy. Do they
Yes.
know what the sorting mechanism is? Like how they sort between the...
Not that I know of, Kathy. Somebody might know, but I don't know what it is. I assume that it's something, they've got a pair of large appendages arising from their anterior end around their mouth. And that may be a set of appendages that has ciliary tracts to either direct particles to the tube or allow them to enter the mouth.
So they get it all the way there potentially before they decide where it's...
Yeah, they don't seem to be selective when it comes to picking up particles.
Right.
So it
Oh,
must
cool.
be at the mouth level.
Bill Honchmaker down in Oregon commented that they had several of these terabellids in a touch tank at Hatfield Marine Science Center, and they must have colonized there as larvae. Is that possible?
You said they must have because you only started off with one and then you had several?
No, actually, it was a concrete tank. We have live seawater coming through, but it goes through a sand filter, which eliminates a lot of things. But we honestly had some really amazing things colonize the touch tank just from the water that was coming in.
That's interesting, William. At UVic, we have a recirculating system. It's not a flow through, but it does have a sand filter. And even so, there's a lot of things that come in and get populated. So yeah, they do have a larval stage. So they must have, the larvae must have come originally from the ocean, because the larvae is too long lived to be repopulating from that existing population that you had in the tank.
Linda Funk says that she loves your commitment to get a good video. The lengths you're willing to take. But she also had a question. She wanted to know, how long does it take to build the tube?
Oh, you know what? Not that long. If you've got one, you've taken it out of its tube and you put it in a C-tape, you know, by the next couple of days, they have built their a tube for themselves.
Oh.
Yeah, so they get right to it and get the job done. So I have one more video. Now it's 7 minutes long. It's the feather duster worm. That's going to progress probably a little long beyond the end of this meeting, but Woody, can I show it? And anybody that needs to leave can just exit.
Well, we can't miss the feather dusters. If you...
Okay, well then I'll show the feather duster worm.
And I think we started a bit late too, so.
We did.
Okay. So the sabellid. Yeah, I like this one.
I've now shown you how two groups of sedentary annelids capture and ingest sediment, in other words, deposit feeding. But other sedentarians have become specialized for suspension feeding. They feed on phytoplankton, zooplankton, and/or suspended organic particles. So I'll show you how suspension feeding is accomplished by members of the family Sabellidae, commonly known as fan worms or feather duster worms. These worms feed on phytoplankton. Sabellids live in a tough proteinaceous tube that the worms secrete and which is firmly anchored to a solid substrate. This is an image of a large cluster of sabellid worm tubes photographed from an intertidal site at low tide. The worms typically live in clusters like this, and all the tubes here are projecting outward from a basal anchorage deep within the rock crevice. When submerged in seawater, the worms extend a delicate plume of feeding tentacles from the open end of their tube. This is a member of the genus Schizobranchia. Its tentacles may be a beautiful maroon color like this specimen, or they may be light yellow to brown. A closer look at the feeding tentacles shows that each consists of a main stem, although in Schizobranchia, the main stems are dendritically branched, and each stem bears a row of parallel side branches projecting from either side. The tentacles of feather duster worms are collectively called radioles, and the side branches are pinnules. So I'm showing you the radioles at increasing magnification. The image of this worm is being recorded with the camera mounted on a dissecting microscope. The radioles, including the pinnules, are ciliated. One tract of cilia creates a flow of water through the radioles and pinnules. Another tract flicks particles onto the frontal surface of the pinnules, and yet another carries the captured particles along the frontal surface of the pinnules and down the main stems of the radioles to the mouth at the center of the radioles. This video was captured with the camera mounted on a compound light microscope and focused on a few pinnules mounted in a drop of seawater on a microscope slide. It shows you the cilia beating along the pinnules, specifically the cilia that deflect particles onto the frontal surface of each pinnule. As you can imagine, these delicate radioles projecting out of the tube are potentially highly vulnerable to predators. So if a sabellid detects an unusual vibration or a shadow looming over it that might indicate an approaching predator, they can very rapidly withdraw the plume of radioles into their tube. So how do they detect a sudden drop in light intensity when their head, where eyes are normally located in annelids, is inside the tube? They actually have little black eyes distributed all along the main stems of the radioles. And here's a row of black eyes along a radiole of a specimen with brown radioles. Remarkably, these eyes are compound eyes. They consist of many individual light-gathering ocelli, each with its own light-focusing lens, like the compound eyes of arthropods. You can see the individual lenses in this image. These eyes probably can't form an image, but compound eyes are very good at detecting movement. So, how does the duster worm project its crown of radioles out of the open end of its tube? To answer this, I played the same trick on this feather duster worm as I did on the lugworm. I coerced it into a glass pipette, and you can see its little peripodia walking up the inside of the pipette to project the radioles out the end. The trouble is, the glass pipette is slippier than its normal proteinaceous tube. Under a dissecting scope, you can see that the notopodial lobe of each peripodium has a long fan of chaetae, whereas the neuropodial lobe is a slit with raised rims bearing minute chaetae called uncini. This is convergent morphology with the peripodia of lugworms that live in J-shaped burrows. The notopodial chaetae fan do the walking, and the neuropodial uncini are tube grippers. But this is kind of mind-blowing, but partway down the length of the worm, the notopodial chaetae fan becomes ventral, and the neuropodial slits with uncini become dorsal. The peripodia reverse their dorsal-ventral identity. So bizarre. This bizarre switch in dorsal-ventral polarity by the peripodia corresponds with another reversal of sorts. Let me explain. Feather duster worms live in a tube that's closed at the posterior end, and their anus is at the posterior end of the worm's body. So to prevent the tube from becoming filled up with feces, they have a so-called ventral fecal groove, indicated here by the arrow. The feces up to the anterior end where it can be discharged from the open end of the tube. And yet, at the point where the peripodia switch dorsal-ventral polarity, the ventral fecal groove moves up the right side and switches to the dorsal side, and it continues to the anterior end and the open end of the tube as a mid-dorsal fecal groove. Finally, how does the feather duster worm secrete its proteinaceous tube? There's a gland at the ventral side of the base of the radioles that secretes the tube material, initially as a liquid. The liquid flows under a collar of tissue, and the collar folds over the peripheral rim of the pre-existing tube. As the new tube material hardens, it's molded by the collar over the rim of the pre-existing tube. The collar is indicated by the arrows, although this worm has been removed from its tube.
Okay, so that's the wonderful feather duster worms with their reversal partway down their body in dorsal ventral polarity. Why? Who knows? Any other questions?
We've got a question from Justin about the cilia.
Yes, Justin.
You want to talk, Justin?
Unmute yourself.
There we go. How's it going? Thanks. So I noticed the cilia movement happens almost on its own function. Obviously, these creatures don't have like a brain. Do they have a brain?
Oh, they do. Yes, they definitely do.
But does it happen as like a primary function or a secondary function?
So Are you asking me, so you're asking me whether these cilia just automatically beat or what
Yes.
controls the cilia? Yeah, the ciliated cells are innervated. And generally things that have ciliated epithelia, the cilia beat autonomously. What they have in terms of neural control is a nerve impulses causes the arrest of the cilia. But they can also
Okay.
have neural control in speeding up or slowing down the cilia. But primarily the neural control is to arrest the cilia. And then once that arrest is ended, they take up their automatic beating again.
Okay, interesting. Thank you.
Yeah, they have a nervous system. I mean, when they detect, for example, a shadow over top of them, so those little eyes, that information has to be communicated to the longitudinal muscles in the body. And so that requires neural innovation, and that's all coordinated within a brain.
No, that's really cool. I see these things all the time. I'm a diver in the Canadian Navy, just in Esquimalt Harbor here. I see these things all the time on pilings, and I just thought they were some sort of anemone. So that was really cool to see that, hey, these are actually worms. So that's great.
You can tell all your friends all about them. I think that's the greatest thing about learning about invertebrates is you can tell all your friends in your family cool stories.
I tried to recruit as many people as I could from my section to come here and watch this, and they just think I'm the biggest nerd right now, but they're lost, you know?
Yeah, they're lost. Absolutely.
Justin, you found your people. You found your people. This is your tribe, my friend.
Oh, I'm the biggest closet marine biologist, you know, apprentice there ever is. I'm fascinated by polychaetes, and somebody is going on their dive course and posted a picture. Apparently these things come up to mate. I'm like, oh yeah. And they come up, we have a huge light that shines into the water, and these things just come up and freak the hell out of all the other divers because nobody really knows what they are.
Yeah.
So I just saw this, someone sent me a link. I'm like, great, signed up for it. Amazing. Anyway.
Yeah.
Sorry, I won't take any of their time.
We have a conference coming this summer, mostly online, but there will be some, I think.
There'll be about half and half, like some in-person field trips and then online conference sessions.
Yeah.
So Kathy, is this the one that you asked me if I might present something there?
Yeah, it's the week of the 15th to the 21st of August. And Justin, there will be field trips both on the island and on the mainland, as long as we're allowed to congregate outdoors as well.
And there will be worms.
Oh yeah.
And Louise will be talking again.
Well, yeah, I'm trying to decide what to talk about. I'm considering doing a chitin dissection, or would you folks rather see a bunch of videos of animal behavior?
Animal behavior.
What kind of animals?
Yeah.
Well, marine invertebrates, you know, snails.
Animals that count.
I mean, chitins don't do much in real time. I was so excited the first time I went to the tropics, I could actually see a chitin crawling in real time, because around here they don't, you have to have a time-lapse camera.
I mean,
we did have one more question, Louise. It was, why are there so many things growing on their tubes?
Oh gosh, worm tubes can be a real nursery for other organisms. So we were talking about the larvae. Many marine invertebrates have a pelagic larval stage, and they're looking for a place to settle. And worm tubes seem to be a very attractive site, place for many species to settle. You know, and as I showed you, these worms typically live in a dense cluster. So for a little juvenile to be down in that cluster of worm tubes, that's a pretty protected place to live, and may facilitate the early survival before they grow to larger size.
Cool. Thank you. Did anybody else have any more questions?
Can I ask one more question?
Yeah.
Absolutely.
So I remember in the labs at Camosun College, they used to take the feather duster worms out of their tubes so they could show some of the stuff you were showing. And Linda at that time commented that she always had to keep track of which tube went with which animal, because they would not crawl back into a tube that wasn't theirs. Do you know, do they have a way of determining which is their tube, do you think? Is there some
Well, you
kind of
just told us that they do, Kathy.
they do, but do you know what, like, do you know, is there a neural mechanism or some kind of a, what, do you know what?
Way
Probably-
beyond my area of expertise. But
probably
there again,
just like a
these animals
glove.
do have a nervous system,
Yeah.
and it might not look like they're doing much, but they are probably a lot more aware of things than we realize.
I always wondered if there might be some kind of pheromone or smell or something that went with the tube that they could detect, but.
Oh, nice.
I
Or
don't know.
maybe, the tube diameter has to be appropriate for their body, right? For the
That's true.
size of body that they have.
Yeah,
Possibly they're looking for that right-sided tube, and no surprise, the tube that they got pulled out of is just right in size.
Right. We did just have another, just a comment that said they have also observed lugworms by putting them between two pieces of glass, similar to an ant farm. Wow.
Right. Yep, they will do that, everting their pharynx in that kind of a situation too. Another thing people have done is get little, you can get little pellets, little beads of transparent plastic, and you can fill an aquarium with these beads of transparent plastic. And that's another way of seeing what animals might be doing. You can kind of see through the beads to a certain extent, but there's lots of interference, change in light refraction looking through beads. So that's not
Mhm.
perfect, but there is various.
It's more of a natural environment for them if they're
Yeah.
being in like sand or pebbles or something.
Or a pipette.
Okay. Yeah.
Yeah.
Well, thank you guys so much. Again, our next presentation is on May 10th, and that will be
On seabed mining.
seabed mining.
Uh-huh.
And yeah, we look forward to seeing you guys there. Louise, do you want to put your email in the chat just in case anybody wants to contact or ask you questions afterwards?
Yeah,
I can
please-
also
please
add
do.
it to the web page.
What would you prefer, Louise?
Well, I've just typed it in.
Okay.
All right.
So I've got a question generally about how this happens, because it seems sort of by chance that I stumbled upon this lecture. Is there a Facebook group that you guys are all a part of, and that these meetings get thrown out onto there, and then everyone that's a part of that will get a notification?
Yeah.
Something like that?
We are the Northwest Aquatic and Marine Educators. So we're a group of teachers and educators from Oregon, Washington, Alaska, and British Columbia. And we also just have people who are interested. So our webpage is PACNAME, NAME. And it's, we announce all of our, it looks like
Jen's got it in the chat.
and we also have a
There
Facebook.
you
And
go.
so Jen Magnuson does our webpage and our Facebook. She just put a link to it in the chat.
Perfect. Thank you so much.
Encourage you to sign up and become a member. It's only like $25 to be a member. And then you get a discount on the annual conference, which will be coming up this summer.
And then you also get emails.
And then you'll get the email. Yeah. Then you don't have to look at the webpage to find out when the next one happens. You'll get an email reminding you. So, but we'd love it if you wanted to come to another presentation, Justin.
Right. No, it'd be good. It's just now the stars have aligned. I'm actually in a quarantine right now, and I have all the time in the world and right now, so it's cool.
Right on.
Cool to sit down and do this.
Yeah.
Oh yeah, no, it's everyone on the ship's quarantining before they go sail on the ship.
Okay.
Oh,
Well, if you're interested in looking at past talks that we've had over this last year, those are on the webpage as well. The recordings are.
Oh sure, like if you guys have done anything on nudibranchs, that'd be amazing.
Not yet, but we should.
Margie's excited about that.
Yeah, we
Yeah.
could do a nudibranch.
We've got a, I'm associated with the Marine Science Center down here outside of Seattle that has a very active nudibranch team. So I'll see if I can get somebody. That'd be a great talk.
Louise, do you have any clips on nudibranchs?
Yes, I do.
Okay. I'll have to get you scheduled in the fall for a nudibranch.
And nudibranchs and relatives like any of the opistobranchs.
Beautiful.
Wonderful.
All
Okay.
right.
Well,
Thanks,
thank you,
everyone.
everyone.
Thanks, everybody. Have a wonderful rest of your month. We'll see you next month.
Thanks.
Take care.
Good.